Hello viewers, welcome once again to our ongoing series on Mendelian genetics. Today we wish to take a closer look at another example of the deviations from Mendelism, which took us beyond the inheritance pattern of Mendelian principles. These type of interactions may appear to be quite confusing because their F1 ratios and F2 ratios may come out to be identical, yet the interpretations for these interactions are entirely different. So, today's interaction which we study requires a little introduction of what we had learnt on the previous turn in terms of incomplete dominance and of course the normal dominance, because there is another type of interaction between alleles where the phenotype of a heterozygote is not intermediate between the phenotypes of the homozygotes at all. Rather, the heterozygote would simultaneously express the phenotypes of both the homozygotes. Now, when it is going to express the phenotypes of both the homozygotes, this becomes a little confusing, especially we can confuse it very easily with what we had learnt on the last turn, that is incomplete dominance. So, let us begin from the incomplete dominance example once again. In case of complete dominance, we have the two alleles which are say A1 and A1, and they are now dominating as far as their phenotypic range is concerned in a cross. If all the progeny are red, then it is obvious that red is dominant to white, and if all of them were to be white in the F1 generation, obviously the conclusion is that white is dominant to red. So, therefore, there is no confusion on this score at least. But then when we are coming to incomplete dominance, then we find that the phenotype of the heterozygote is going to fall say somewhere in between the phenotypes of the two homozygotes, and the degree of having an incompleteness may be variable, and this is what we had discussed on the last turn as to what is the threshold that a particular allele alone would be able to accomplish. Now, this diagram puts a very obvious question in our minds. Why is this not an example of codominance? Why only incomplete dominance as we are asserting? The point is that the flowers would exhibit codominance only when the heterozygote produced both red and white pigments, and which then would combine to produce a pink phenotype. Now, what would be again incomplete dominance? That is, the heterozygote produces only a red pigment. The pink phenotype comes about because the amount of pigment produced by the heterozygote is obviously less and much less than the amount which was being produced by a homozygote, and that would be incomplete dominance. So, that means it gave us a very false idea of say a near blending, which it was not. But then in case of dominance, it was clear that we had discussed that there is no question, it is only one type of phenotype. In incomplete dominance, it is intermediate, but then in case of codominance, the phenotype of the heterozygote includes phenotypes of both homozygotes. If I was to give a very crude analogy of this whole controversy, it would be that incomplete dominance means a mixture, a near mixture or intermediate of both, whereas codominance means this as well as that. So, that means when this is being crossed with that, both of them have an equal say. Let us take a few examples to illustrate the point. We take the first example of Shorthorn cattle. Now, in this case, we know that the cattle have different coat colors. So, the homozygous red would be represented by capital R and capital R, whereas the homozygous white would be with capital W and capital W. A mating of the two would have a progeny whom we would call as a roan. Now, this would not be a mixture of red and white in terms of pink. It would be red and white. In other words, it could either be white with red patches, or it could be opposite, that is red with white patches or white with red patches. So, that means this roan is in fact heterozygous, which represents the characters of both the parents. Likewise, a homozygous red bull and a homozygous white cow would produce a roan, that is a spotted offspring. Let us look at the checkerboards of these crosses. If we were to cross pure red bull with a pure white cow, now we end up with a roan, that is all the members of the F1 generation are roans. Now, here you would signify that capital R and capital W, both of them are expressing themselves even when they are together in a heterozygous form. So, this forms the basis of codominance. Likewise, if in the next generation both the roans were to be bred, then we would find obviously the ratio being 1:2:1, that is 1 pure red, 2 roans, that is 50% red, and 1 pure white. So, in this case also we need to have a new set of symbols which are to signify the codominance, and this idea was given by Stanfield and Suzuki and co-workers. Now, they say that for all the codominant alleles, all the uppercase base symbols with different superscripts should be used. As we had been using R and W together, they say no, it is of a different type. So, that means the uppercase letters would indicate that each of the allele would express itself to some degree, as well as the other allele. So, that means they are going to since they are going to express themselves in equal measure, we have say something like C to the power R and C to the power W, and we have been repeatedly saying that it will be better that if we represent these alleles say in itals, because that is now the convention. So, according to the new convention, now the breeding pattern that we see for these cows, we also modify the notations accordingly. Here is another cross between two Shorthorn roans, that means both the parents are heterozygous, and then we get again 1:2:1 ratio, that is 1 red and 50%, that is 2/4 roans and 1 pure yellow. Next, we take an example of horses. It will be of interest for you to know that there are about 49 different colors of horses. Of course, they have color which the breeders at the stud farms, they are always interested in modifying these coats for better breeding purposes and for better market value. And one example of these is the famous Appaloosa horse. Now, this is actually a combination of a grey and a white horse. Now, the grey horse would have the alleles capital G in a homozygous form, and the white would have WW. In their mating, we get a heterozygous horse, which is showing say capital T GW, and this Appaloosa is a white horse with a grey spot. So, that means the Appaloosa horses are generally bred for their unique texture of their hair and color. Likewise, we have another example. This is a common household example that we see even around us, the speckled chickens. Now, some of these poultry have absolutely black feathers, the other ones they have white. But then a combination of the two is also a good example of codominance. We have black and white together, what is called as speckled feathers, which are represented by the genotype capital B, capital W. One very important point which we have to remember, and that is each of the feather would not be black and white. The feather would either be black or it would be white. So, again we keep on reiterating that there is absolutely no blending. Codominance manifests itself in plants too, and we have the beautiful flowers of rhododendron, which belongs to the family Ericaceae. And here we find that the red allele, which is controlling the red pigment, and W for white, a cross between the two homozygous character parents would now end up in all the progeny which have white as well as red flowers and beautiful ornamentations, beautiful complexes of white and red. A study of codominance without the Mn blood groups in humans seems to be absolutely incomplete. Now, this is a blood group which is not as prevalent as ABO group, which we are going to study subsequently, but then again it is very important because it also occurs in some of the populations of the world. Karl Landsteiner and Phillips Levin, they were the people who observed that there is the presence of two distinct molecules, and obviously they are glycoproteins. They are located on the surface of the RBCs, and it is by virtue of these 2M and N that these particular antigens are going to provide the right biochemical and immunological identity to the individuals. We agree that it is less clinically important, yet it forms a very good example. So that means a person can be having an RBC with MM, capital M, or capital NN. What is important is that if an individual has capital M, capital N, then he or she doesn't show an intermediate type. This is a different type altogether, which again illustrates the example of codominance. So that means this is entirely a new genotype or a third genotype. So in a human population, we find that either we have M or N, and they are controlled by the alleles. Capital L is in honor of Landsteiner, LM and LN for controlling these antigens. Incidentally, both these genes are located on our chromosome number 4, and what we find is that there is a proper serological pattern which the M and N groups follow. For example, the anti-M is going to react with the LM, and this is how we find out which blood group it is, and likewise the LN and LN is going to have a reaction with anti-N, but then the new genotype, that is LM and LN, is going to react with both, that is anti-M as well as anti-N. This is a similar type of procedure that we find in the ABA groups also. Now, mating between two parents which are having MN group, that is both of them are heterozygous, would again end in 1:4 is to 1:2 or 1:4, that is 1:2:1 of pure M, pure N, and the maximum would be the MN. We have another example of a cat who has a mosaic type of coloration in a calico cat. And you know that the bar bodies were first discovered in cat only, and so some of the alleles which are present on the bar bodies, they are inactivated, but the alleles which are present on, say, the other chromosomes which are not heterochromatized, then they are responsible for showing codominance. Just by the way, I have shown deliberately a mouse along with the cat. This gives us a very good idea of Tom and Jerry, and we know that all our sympathies are always with Jerry. Now, this is not unjustified, because recent studies have shown that we are nearer to a rat than a cat. Now, this is a matter of synteny, which we are going to follow up when we start the series on molecular genetics. But then they have a lot of genes which are and the sequences which are common. Coming back to the promise that I had made in the last lecture, that we will be talking about sickle cell anemia in greater details, because it is more than just codominance. In fact, it is an illustration of so many examples of the principles of Mendelism and beyond Mendelism. Now, sickle cell anemia is a disease which is very prevalent in Africa. We also find that the population of African Americans also have this gene. They are carriers, so we say that they are the sickle cell carriers. Now, actually, this particular disease is caused by an abnormal hemoglobin, and it is abbreviated as SCD. And here the RBCs, they become sickle shaped rather than being biconcave. I have just included a sickle which is there, which is used for harvesting, just to give an idea of how the topology of a sickle cell RBC looks like. Now, this has a lot of problem as far as the shape is concerned, because it would have lot of problems in moving through the blood vessels. It becomes stiffy, it is sticky, and therefore the blood vessels can be clogged. And when the blood vessels are clogged, there is a blocking of the blood flow, and it results in serious infections. In addition, the RBCs which are normal would have a normal lifespan, say, of 120 days, but the sickle cells would have only, say, 30 days. Even the oxygen carrying capacity is reduced substantially. So that means everywhere we have a problem with the sickle cells. So three people, James, Herrick, Hahn and Gillespie, as early as in 1927, they gave an idea that this SCD was in fact due to a mutation, and it was heritable, but under no circumstances it was infectious. Now, in the 1950s, the famous scientist Ingram, he also studied the amino acid sequence. He modified the Sanger's technique, and he found out that yes, there was in fact a difference in the amino acid sequence. Just to give you a little recap of hemoglobin, hemoglobin itself is a multi-polypeptide protein. That is, it has 4 polypeptide chains. There are 2 identical alpha chains which are having, say, 140 to 141 amino acids each, and likewise 2 identical beta chains of 146 amino acid each. Now, each of them has a characteristic amino acid sequence, and incidentally they are controlled by the genes which are present on different chromosomes. For example, the gene for beta is located on our chromosome number 11, whereas the alpha is on chromosome number 16. And just one, I repeat, just one minor change or modification in one of the amino acids in one of the chains is responsible for playing havoc and converting a normal RBC into a sickle cell one. And it is just a case of transversion or a missense mutation. As you would see on the screens, we have the 11th chromosome, and one of the chains of alpha in the 6th position was to code for glutamic acid, and the sequence was GAG. Now, a change occurred in the A part of GAG, converting it into GTG. So, it was a conversion from A to T, that is from a purine to a pyrimidine, which changed the entire sense. So, this missense mutation is what we call as transversion. Had it been transition, that it would have been a purine to purine or pyrimidine to pyrimidine interconversion, and as a result the polypeptide now starts behaving abnormally. It starts behaving abnormally. Now, some of the recent ideas have gone even beyond that. we now appreciate that this particular gene for hemoglobin is in fact belonging to a multigene family, which is a recent concept. So that means this is a gene which can it has multiple copies and it can function at different stages of development. What do we mean by this? What we mean is that many of the copies can be turned off and on in a sequential manner, either in an embryonic stage, in a fetal stage, or even in an adult. So that means the manifestation of SCD can be different in different stages, and it may even say lead to lethality if it is combined with other ailments. But the point here which is important is that whenever we are going to have, say, gene therapy, one has to look for which type of the aspects of these genes are active at which stage of human development. So coming to the genetics once again, we have the three genotypes which are having different phenotypes. HbA is normal RBC, no sickling at all. HbS doesn't show any anemia, but then these RBCs would be sickled only under hypoxia conditions. That means when there is a low oxygen concentration. And in case of HbS and HbS, there is going to be severe anemia, and this cross which we find with the sickle cell anemia would now give an inheritance of sickle cell disease. Two parents who are the carriers could produce one that out of 4 normal children, 2 carriers and 1 sickle cell anemic. What we wish to drive home here is that as far as anemia is concerned, HbA allele is dominant. As far as RBC shape is concerned, it shows incomplete dominance, because in HbA, HbS we find that there is a sickle cell trait, and whereas the hemoglobin is concerned, it shows codominance. And when the two HbS and HbS are together, then obviously it means it is a case of lethality. So the example of SCD beautifully illustrates in fact the Mendelian as well as the non-Mendelian genetics all together. And we find that this type of disease is or the allele frequency is more than 20% in populations in Africa. The one last point which one should remember here is that surprisingly HbA, HbS people are resistant to malaria, and this is an example of what we call as the heterozygote advantage. In other words, there is some goodness in this misery also, that is the people who are sickle cell trait possessing, they would not be having any problems with the Plasmodium vivax. Now in nature there is a beautiful balance of having problems with the heterozygotes as well as the advantage of having immunity against a particular disease. This is what we call as balanced evolution. Thank you.